immunity to parasitic infections either will lead to a lack of an immune response. In other words, the body is totally unable to tell that there is an infection going on. In other words, some parasites have really evolved so that they are able to get into the body and the body doesn't detect that there's anything going on. And if that happens, then of course there would be a super infection. There would be a very overwhelming infection in the body because the parasite has evolved to manners and to ways in which there's no way the body is able to detect. So that, of course, causes what we call super infection. And that might eventually be detected and cause the body to over-exaggerate in how it responds to the infection ultimately. And if that happens, then of course, sometimes it can be life-threatening. So we look at specific ways, but so of course, this would lead to say sometimes persistent infections of these parasites and what we would refer to as chronic infections, chronic parasite infections. And that of course has its own damages that it does to the body. So it's important for us then to look at specific examples of how these parasites behave. For instance, there's been an observation, especially in the late 80s, where there was something called immune complex induced nephrotic syndrome in Nigerian children. So this is just malaria, but this malaria is not caused by Plasmodium falciparum. This malaria is caused by Plasmodium malaria. You know, if you have any idea about, I'm sure you're doing in microbiology now, if anything, but the most notorious parasite, of course, that causes malaria is Plasmodium falciparum. That is what 96% of the cases of malaria are caused by this. So we have other, of course, plasmodial agents, other four, apart from Plasmodium falciparum. We have malaria, we have ovale, we have vivax, and of course knowlesi, which is both in non-human primates as well as humans present there. So they've been characterized in humans and they are responsible for causing malaria. Now in Nigeria, there was a discovery of children who had issues to do with their kidneys. Of course, they would present with swollen facial tissues. They would present with anemia. They would present with respiratory distress. They would present with quite a number of issues. And when this was being checked, every 4 hours, they would end up having fever. Every 4 days, sorry, they will end up having fever. And of course, cotton malaria, this is malaria caused by Plasmodium malaria, is characterized by that. These individuals usually end up having 4 days. Every about 72 hours, they end up having fevers. So by the time this was being discovered, their kidneys had really been damaged. And you see, so the parasite was able to invade the body. The immune system was not able to pick it. There were super infections. By the time it's being discovered, the kidneys of these children are already damaged. And that's the trouble with parasites. Of course, partly liver can be damaged sometimes when you schistosomiasis. So schistosomiasis, of course, is another causative agent, it's another parasite. What it does, it triggers production of interleukin-4. This is a cytokine. And this interleukin-4 mediates formation of a granuloma. Now, a granuloma is a giant macrophage. It's a giant macrophage that traps these eggs. So these parasites survive by laying eggs. And of course, once the eggs are laid, they're able to be hatched and the parasite continues thriving in the body, especially in the liver. So When the liver cells discover this, they try to form, of course, granulomas. And what we call by granulomas is a merger of macrophages. We know what macrophages are. They are monocytes that moved to the tissues and they became resident to the tissues. Once they were resident to the tissues, they are phagocytic, of course. And these are large cells. So what happens is that these macrophages they merge together with the help of interleukin-4 so that then they are able to trap whatever it is. For example, the entire population, especially in Africa, a third of the world's population is said that we have TB. Of course, most of you would be having TB in your bodies. Definitely have it. But it's in latent TB. It has not yet been fully expressed. Now, if you get to a situation where you are immunocompromised, maybe you have an immunodeficiency that is being caused either by some chemotherapy, some cancer there, or HIV, or you are just born with immunodeficiency in general. What will happen is that tuberculosis will now manifest. Maybe it's going to end up being pulmonary tuberculosis. The reason why you have latent TB is because of these things we are calling granuloma. The granulomas are large macrophages that trap the causative agent. So the causative agent can't move. In this case, we are talking about schistosoma cells, but it's also possible to trap bacteria like Mycobacterium tuberculosis. It's possible to trap that. Are trapped, then they can't be released. They are only released especially when the body, the immune system or your immune schistosoma mansoni or schistosoma, especially schistosoma mansoni, which is the main causative agent of schistosomiasis. The eggs that are laid usually trigger production of interleukin-4, and this interleukin-4 forms a granuloma. And this granuloma is being formed in the liver, and that can cause liver damage. And of course, with liver damage, then you know how important the liver is. So this is a parasitic infection that can cause death. You just end up having liver cirrhosis because of a parasitic infection. Then, of course, parasitic infections can sometimes be even trigger autoimmunity. We have an idea of what autoimmunity is. We'll discuss a whole topic about autoimmunity. We're going to, maybe 2 lectures to come, we're going to discuss autoimmunity in a bit of details. But we have cross-reaction between the parasite and self-antigens. So parasite antigens as well as your body antigens can cross-react, and this would lead to autoimmunity. An example is the Chagas disease. The Chagas disease is caused by another parasite called Trypanosoma cruzi. Trypanosoma cruzi is what causes Chagas disease. So Chagas disease, these individuals end up having cardiac issues. They have cardiomyopathy. You are infected with trypanosoma. Of course, trypanosoma causes trypanosomiasis. It's a parasite, especially in regions, especially that have large water bodies. So it's a parasite that thrives there. This parasite survives in snails, but snails are intermediate hosts. Human beings are, of course, the definitive hosts. So individuals that end up having trypanosoma, we have quite a number of types of trypanosoma, and the Chagas disease is caused by specifically Trypanosoma cruzi. There's an example here down below here that is Trypanosoma brucei. This is different. The Chagas disease is caused by Trypanosoma cruzi. So individuals with this disease, and especially is present, it's common in the Southern America, in the Americas. It's present there mostly, those regions. So these individuals end up having heart issues. And what I'm saying, there is no single way of dealing with parasitic infections. Each parasite has its own challenges. So we're just looking at a parasite here, a parasite there, what happened here, what happened there, what happened there, what our papers are reporting. Of course, we expect antibodies, we expect infection by parasites to lead to antibody production, because antibodies are now supposed to neutralize what? The antigens. And those antigens expressed by these parasites on their cell surfaces. So some of the immune responses, especially antibody immune responses, are very effective against some parasites. For example, Trypanosoma brucei. These are just like malaria parasites, they undergo stages. For instance, a malaria parasite will form a sporozoid, will form a merozoid. I want to believe that is being covered in your microbiology, if not. So the antibodies can be generated against some specific blood parasites like Trypanosoma brucei. And once that happens, then they're able to eliminate these stages. For instance, the sporozoid as well as merozoids, even those of malaria. Trypanosoma brucei is also able to undergo these stages, some of these stages, the sporozoid, the merozoid stages. Now, that is part of the reasons as to why parasites have issues. The immune system cannot handle parasites very effectively because, for example, if you are to take malaria, malaria has two cycles in the body. There is the hepatic cycle, there is the erythrocytic cycle. So as these cycles are going on, the parasite is changing from, say, a sporozoid to a merozoid to a schizoid. So does the immune system target antigens against sporozoids? Does it target antigens against merozoids? Does it target antigens against schizoids? Does it target, so it is confusing to the immune system. But the reason why, for instance, antibodies are very effective against sporozoids and merozoids in this case is because they are blood parasites. Even if it's malaria, this is a situation that is happening in the erythrocytic cycle. But now the body can be unable to detect the schizoids, for instance, because schizoids are now in the hepatic stage, they're in the liver. So it's a bit sometimes confusing to the immune system, but at least in this case, the immune system is able to produce antibodies against blood stages effectively for the case of, say, malaria and for the case of Trypanosoma brucei. It's also possible to produce enough eosinophils. We know eosinophils are a type of white blood cells, and IgE. IgE, we know that IgE is a type of immunoglobulin, of course, especially affecting the respiratory system. So eosinophils as well as IgEs are very crucial in eliminating a parasite called Trichinella spiralis. So Trichinella spiralis can be very easily dealt with because the body is able to produce enough eosinophils. It's also able to produce enough antibodies, that is IgE, be able to then eliminate this particular parasite. Let me admit this first. Okay. Then again, the immune system is able to modulate the immune response. For instance, production of anti-IL-4 and anti-IL-5. Anti-IL-4 reduces IgE production. Okay. Anti-IL-5 suppresses eosinophilia. You see here, we said, for instance, Trichinella spiralis can be dealt very easily with by the use of eosinophils, which are a type of white blood cells, and of course by production of IgE. And we know that for IgE to be produced, we require interleukin-4. Interleukin-4 is quite important. Interleukin-4 is also still the same cytokine that has been involved, especially in granuloma formation, where there is merging of macrophages. We just pointed that a few minutes ago. So if you produce anti-IL-4, what happens is that IgE production is reduced. If IgE production is reduced, then you find animals being infected with helminths. And of course, eosinophils are also very effective against helminths. Helminths are what we call the worms. So I'm sure if you defined what parasites are either protozoa or metazoa. So protozoa are the likes of the trypanosoma, the malaria, the trichinella, while helminths, the metazoa, which are the worms, are the partly of helminths. You can divide parasitology into, say, protozoology and helminthology. So helminthology, of course, is where you're dealing with worms. And worms are usually effectively dealt with IgE and eosinophils as well as basophils. Basophils are also effective against worms. Now, you remember when we were discussing previously, I said for individuals especially who end up having these asthmatic attacks or hypersensitivity, what is causing that asthmatic attack is overproduction of IgE, And I said, if an animal or you are infested with helminths, that is worms, the IgE levels in your blood are so high compared to an individual who has asthma. And I said partly, why is it that an individual who has worm infestation does not die, but if you took the same titer, the same level of IgE levels and took that to an individual who has asthma, they are going to definitely die of that. Part of the reasons is that the body is able to produce T regulatory cells. I think I said that. It's able to produce interleukin 10, and interleukin 10 are partly what we are referring to as anti-IL-4 and anti-IL-5. They suppress the body so that then they're able to control this body so that as much as there's high IgE levels, you don't go into respiratory issues. You don't go into bronchoconstriction, and you don't end up dying because of helminths have to enable you to survive also. They have found a host. So if they kill the host, then how are they going to survive? So they've evolved to a level whereby they will cause damage to you, but will also ensure that you're still living by causing the immune system to auto-regulate itself so that, again, you don't die. Now, so of course, this is being tried out as the solution to asthma. Of course, this is being tried out as a solution to asthma. I think this experiment has been going on for years now. It's over almost 30 years, but I don't know. There's never been. It's not very promising. We don't have very promising results. Just have to understand how helminths are able to survive in the body with those high IgE levels without killing the host. So not so much is coming out as new information currently. So you are still stuck there. But of course, if we're able to break this, then we'll get very good solutions, especially for people who have asthmatic attacks and people who have, say, rhinitis. It's very possible to find a very good solution from this. Now, we have also schistosomosis or schistosoma. Schistosomiasis, of course, is another condition that is parasitic. So how does the body deal with schistosomiasis? Now, so we have immature schistosomes that are called schistostomules, and they can be killed by antibody-dependent cell cytotoxicity. That is what can kill them. So antibody-dependent cell cytotoxicity is a type of hypersensitivity that we'll discuss when we'll be talking about hypersensitivity. But what is happening here is that an antibody called IgE is generated. And when IgE is generated, this IgG, not IgE. IgE is for respiratory reaction. IgG is being generated together with eosinophils. But IgG in this case will act as an opsonium. So it coats it coats the schistosomes or the schistosoma. It coats it. Once it's been coated, then cytotoxic cells, and we have an idea of what cytotoxic cells are. Maybe can somebody remind us some of the cytotoxic cells we've discussed before? For number, I think we have like two we've discussed. One in the innate system, one in the adaptive. Cytotoxic cells, cells that are able to perform cytotoxicity. Can anybody remind us some of these cells? They're basically two. Anybody to remind us these cytotoxic cells?
CD8.
CD8, okay, CD8, a T cell. Any other? Any other cytotoxic cells? We said there's one that is innate. You remember we talked about innate cells being neutrophils, macrophages, and this other type of cell that I want, if you remember.
The natural killer cells.
Correct. Micah named them correctly. So natural killer. So natural killer cells, of course, are the ones that are brought in this case. And these natural killer cells are able to perform cytotoxicity by granzyme. They use granzyme and perforin. Those two proteins are able then to, of course, perforate the cell and kill. They're able to effectively eliminate schistosoma, schistosomiasis. Then we have intracellular parasites. Intracellular parasites. An example of these intracellular parasites are like the Toxoplasma gondii. I don't know. I'm not so sure how far you people have you finished? Are you currently doing microbiology?
Yes.
So you've come across some of this terminology, the parasites you're talking about? Ama bado mnafanya bacteriology?
Bado tuko hapo bado tujafika hapa. Lakini gondii we've heard of gondii.
You've heard of gondii, a name of an individual or a name of a parasite. Okay. So, all right. So, watu wenye wanapenda paka sana, this cats are dangerous animals. So most cats, of course, are going to be hosts. If maybe by the time you're doing parasitology, you'll discover that we have what we call intermediate hosts and then definitive hosts. Intermediate hosts, then definitive hosts, especially for parasites that are zoonotic. Zoonotic or zoonosis is where a parasite is able to maybe infect an animal and ultimately infect a human. Now, Toxoplasma gondii, very, very present in cats, but can also infect human beings. Let it find you when you have a failed immune system, especially maybe a HIV patient, a cancer patient, a person maybe who's using drugs that are really causing damage to immune cells. It can be lethal, can be very lethal. Sometimes it's a quiet parasite, can be in the body, nothing is happening. But the moment you become immunocompromised, then you realize it's a very dangerous parasite. We've also talked about Trypanosoma cruzi. I think I've already talked about this in what we call the Chagas disease. Autoimmunity can be caused by this. So it's an intracellular parasite, which means they get into the cell. Leishmaniasis, Leishmania, all types of Leishmania species are also able to get into the cell. So they're able to evade macrophage killing systems. They're able to evade macrophage killing systems. And so what happens is that cytokine production by T cells Of course, this triggers, we realize that Th1, of course, overproduces or secretes interferon gamma, which is crucial for macrophage activation and the elimination of intracellular parasites. So, of course, this should be done by a cell-mediated immunity, but these parasites can very easily evade because they will hide in the cells, they'll hide in the cells, they will not even express their antigens. Once they are there, for example, you remember when you were talking about Trypanosoma cruzi, there is super infection to a level where even the heart is affected. So you find that they are producing, yes, their antibodies, but it's like they coat the antibodies with human proteins, kind of. So you find that there is a cross reaction between the antibodies of the parasite and self-antibodies. And once that happens, then it's possible to even trigger autoimmunity. That has been seen. So intracellular parasites can very effectively evade macrophage activation because that's the possible way of killing. They need to be presented via MHC1, of course, in this case. But what happens? They can't even be traced. Their antigens are hidden. And part of the reason is because they also undergo different stages. So by the time the body has been able to discover this stage, be it a sporozoid, be it a merozoid, they're able to discover this stage, they've already jumped to another stage. And once that happens, of course, the immune system is confused. Leishmaniasis, in the case of leishmaniasis, it is associated with interleukin-4 production. We've seen quite a number of things that interleukin-4 does. Interleukin-4 in granuloma formation. It's also characterized by interferon gamma. So interferon gamma just helps, of course, in macrophage activation, so that then maybe they are eliminated by macrophages, especially neutrophils and neutrophils, as well as macrophages, of course, can be expanded by interferon gamma. Okay, elimination of intracellular parasites, of course, would be done by direct cytotoxicity. It will also be done by, so interferon gamma activates CD8, and this CD8 are able then to cause direct cytotoxicity. So these parasites could involve, of course, malaria, rickettsia, chlamydia. All this can be eliminated by cytotoxicity. We know that intracellular parasites or intracellular pathogens generally can be eliminated by cytotoxicity, but they're able to evade in a case of say macrophage or phagocytosis. Then Th2, of course, Th2 cells the T cells that are Th2 can also play a role in terms of parasite elimination. They're especially important in expelling gastrointestinal worms, what we refer to as Helminths. They can be destroyed, especially when they are still in the larval forms. They can be eliminated by Th2 cytokines. You remember Th2 when we are talking about Th2, what they are, and the cytokines that are producing Th2. Remember what the Th2 cells produce, especially during in their activation. They kind of regulate the immune system, but their main role, of course, is to help B cells in production of antibodies, as we saw. So ectoparasites like ticks. I don't know how ticks are able to invade. Do ticks get to human beings? I don't know if they're just cows or humans. Do humans get infected with ticks? Are ticks able to cause anything, any problem in a human being? Or is it just cows? Okay, so ticks can be, of course, dealt with by Th2 cytokines. They are referred to as ectoparasites. They are mostly outside the body. They are outside the body, but so they can be eliminated. And what happens is that they are able to, Th2 cytokines will trigger the granulation of mast cells. And the components of mast cells then will be able to eliminate what you are calling the ectoparasites. This is just the mechanisms for which you can eliminate we call the nematodes. Nematodes are just worms, how you can eliminate. So of course, nematodes would be found in the gastrointestinal tract. This is where worms are found. And these worms would present their antigens. The antigens these antigens are seen by B cells, and we know that B cells, dendritic cells, macrophages, all behave as antigen presenting cells. So T cells encounter this antigen via antigen presenting cells. When that happens, of course, then T cells again interact with the B cells, and these B cells are able to produce immunoglobulin E and immunoglobulin G. So we know that immunoglobulin E, will trigger an inflammatory response to again ensure production of immunoglobulin G. And this immunoglobulin G, we've seen that is very effective, especially in what we call antibody-dependent cell cytotoxicity. So it will act as a coat, but other than that, T cells are able to generate some other cytokines, and these cytokines will stimulate goblet cells. And if you know, goblet cells line up the gastrointestinal system, and the goblet cells are responsible, of course, for production of mucus. So other than these cytokines triggering the production of, stimulating goblet cells to produce mucus, and IgE coating the nematode so that it undergoes ADCC, this mucosal production will also trigger the body to just, so an individual might undergo diarrhea. The individual might undergo diarrhea, so that then there is expulsion of this nematode from the body. That is exactly what we've been talking about. So the mechanism would be, of course, IgE would pass through the gut, and the IgG would damage the nematode. This damage, of course, will result from IgE-mediated inflammation, because IgE will trigger an inflammatory response. IgG, of course, will respond via ADCC. We've seen what ADCC is. Antibody-dependent cell cytotoxicity is going to happen in that case. Then T cells are activated. These T cells are able to release cytokines. These cytokines stimulate proliferation. As that happens, of course, there is increased gut motility, and it aids the expulsion of the worm. So the worm is then removed. And also mast cells, a type of white blood cells, of course, are also again to be degranulated. As they are degranulated, they contain mediators like leukotriene D4. This leukotriene D4, you remember when you are talking about innate immunity in general, some of the mediators would be like leukotriene D4. And this promotes gut motility. So there's movement, of course, in the gut. And that would lead, of course, to, say, diarrhea by inhibiting glucose-dependent sodium absorption. And mast cells also are able to produce histamine and prostaglandin E, and that will promote, of course, diarrhea. And as diarrhea is happening, then, of course, the nematode is being, or rather the worm is being expelled from the gut to the outside. And system is able to eliminate nematodes in general. Nematode helminths, they're also called metazoa. They are the ones that can affect. All right. So that then, of course, brings us to the end of immunity to parasitic infections. If you are to ask me, there is no proper structure about parasitic infections and how the immune system behaves. It deals with fairly differently from another parasite. So it's a fairly complex infections that are parasitic. Say if it were fungi, the mode and mechanisms are more or less the same, so it's easier to of course explain mechanistically. If it's a bacteria, be it gram positive, be it gram negative, it is fairly easy to explain mechanistically. But for parasites, you see, parasites are a bit complex, as I've already said. They have to undergo life cycles, and these life cycles means that proteins are also changing, meaning the antigens are also changing. So the immune system cannot really deal with each stage conclusively. And that's why parasitic infections, of course, are usually a very big problem. They are a very big problem. Despite various ways of dealing with them, they are quite problematic. Yeah, so the only other way would usually is to eliminate the parasite or the intermittent host of the parasite in the population. So, of course, we've seen very many ways of dealing with parasites, especially for malaria. How many campaigns have you had? Up to this point, I think they've only succeeded to generate how many? 2 vaccines of malaria, and they're only effective in children. Vaccines are not even effective in adults; they're effective in children only. So, parasitic infections are quite a problem. Yeah, and with the advanced technology still, we can't come up with a functional vaccine against malaria and how malaria has devastated the population and killed so many people. So that is how the complexity of parasitic infections and how the immune system deals with. So you'll bear with me, it is just like that. That is how the best we can be able to explain immunity to parasitic infection. There's no single way of how we can then explain that, okay, the mechanism is like this, so this is triggered and this is triggered. It is not that simple. Every parasite behaves differently. And we know that we have so many parasites in nature. These are just but a few. We have quite a number of parasites of medical importance, of course. And so some are common in this part of the world, some are not. And so that is how then we can present it. All right. So I'll assume that we have an idea of how then you can be able to explain immunity to parasitic infections. We have general mechanisms, but we also have specific mechanisms depending on how a parasite, of course, is behaving. So if your parasite lays eggs, most likely it's going to be dealt with by forming what we call a granuloma. If a parasite is in the respiratory system, then definitely you're going to ensure that IgE is produced. If a parasite is in the blood, then IgG will definitely be produced. And we know the sequence. You start first of all with IgM, then there is class switching to IgG, and then we've seen that IgG plays a key role in ADCC, antibody-dependent cell cytotoxicity. So those are some of the general mechanisms. Of course, we know that phagocytosis is possible, although it's not very effective against intracellular parasite, but it's possible. It's an alternative. Yeah, but the most effective way for intracellular parasites has now emerged to be cell cytotoxicity, of course. So either natural killer cells or CD8 T cells are then brought. They generate perforin, they generate granzyme, and you're able to eliminate that particular parasite.